Welcome, everyone, to this episode of the Forefront 5, our podcast. And though when I started the podcast, I promised I'd stay within 5 minutes, this week's guest is so interesting and so smart. I'm not sure that we'll comply to the 5-minute rule. So I'd like to welcome Gus Natale. Gus, how you doing?
Yeah, I'm doing well. Thanks for having me on, Betsy. I appreciate it.
Yeah, so I think, Gus, you've received a ton of emails on COVID testing, as have I. So we thought it was a good topic to discuss. So let's talk about testing for COVID. I want you to kind of outline some basic swabs for patients. We'll cover the patients first. Talk about these swabs, how you actually go about testing patients, and your recommendations as if we dermatologists should be doing those in our office.
Yeah, so the test everybody hears about on the news is actually the PCR test. So that's the test that you're trying to use to diagnose somebody with acute infection. So that's done through a nasopharyngeal swab. So if you haven't seen this done, it's a long, basically Q-tip applicator that gets pushed through the nose back into the nasopharynx. So it's going back, 5 inches, and you're getting a specimen that way. And then it's stuck in various medium, most commonly the VCM medium, and then sent to the lab and run. They have looked at testing just the front of the nose and also the pharynx and the mouth, but those have shown in studies to be less effective than the actual nasopharyngeal swab. So the issue with this swab is that it puts the person performing the swab at considerable aerosolized risk when you're doing that. So when you're seeing people doing these, obtaining these specimens, you'll see them in full PPE gowns, typically a shield that goes over the entire face, N95 mask, gloves. And this is very resource intensive because you have to change that PPE with every patient. So right now we're not recommending that we do these in the outpatient dermatology clinic just because there's a risk to the provider getting infected.
That's great to know. And I know that I'm not wearing the full gown, et cetera, in my clinic. So if I have a patient, where do I send my patient if I think they need a COVID test?
Yeah, so I've found at least 3 places near in my region, but it is tough. It's very regional. I think each physician needs to reach out to their colleagues if they don't know of a place or have quick access to a place. But here in Indianapolis, the Lilly Pharmaceutical Company is offering testing. There's actually a private lab that will take cash from any patient and do testing. But tests aren't as available as you may want. So, your local LabCorp or Quest, a lot of those reagents and lab tests are being pushed into the hospitals for hospitalized patients, ventilated patients, sort of more severe patients, and it's hard sometimes in the community to get testing available for a mildly symptomatic case or certainly an asymptomatic case.
Well, maybe that's something we as the COVID task force could dig in with the regional managers and come up with some clinic by clinic guidelines on where to send patients. So that's a good summary as for an acute COVID suspected patient. What about all this antibody testing? I swear, as does everyone else, that I had COVID at one point, and I'd love to get an antibody test. Let's talk about that. How good are they? Give us a little summary.
Yeah, so the antibody tests that have hit the market, there's one major variety, which is sort of a point of care test where you take drops of blood and it kind of runs through a little meter in the office. That is a way to quantitatively see if someone has been exposed to SARS-CoV-2 or COVID. Those tests they can identify IgM and IgG. There's also coming to market a ELISA test, which is taken from a patient's serum, so it requires a blood draw. And that's a way that you can quantitatively see how much antibody to whatever antigen you're looking at is in the patient's blood. I think a lot of the stuff you've seen on the news is about these sort of qualitative tests that have hit the market. The main issue with those is that the FDA has really relaxed their guidelines on getting a test approved. So all these have been done under the emergency use authority and basically have not been proven efficacy in the standard FDA way that they do things. So the issue there is that a lot of these are hitting the market, and some are probably of dubious quality, and some of this is going to have to be sorted out later. So I think that's one of the concerns that a lot of people have about these tests is you don't really know sort of the quality of the tests, and also a lot of these tests don't have true documented sensitivities and specificities. But some of that is trying to be figured out as we speak.
So that's kind of scary, right? That if you got a result from one of these tests and it changed your behavior and you didn't wear a mask, et cetera, there's consequences.
Yeah, no, I think that's the big issue. So one of these tests, which was put out by Abbott, which was supposed to be one of the quickest tests that like a family practice doctor could do in their office, really received a lot of press. And when they actually, there was a study that came out maybe last week, when they actually looked at the data and compared it to sort of the gold standard, and they're using, most of these tests are using gold standard PCR positivity as the gold standard. But this test had one of the worst sensitivities in the whole group, something like 15% of false negatives. So I mean, think about that. That's a high rate. I mean, I would say anything above 1 or 2% is high, and most of these are going. At least the couple that I've actually seen numbers on have a 5% or greater false negative rate.
Yeah, for sure. So do you think that dermatologists, forefront dermatologists should be getting the right test and testing staff and, the clinicians in the office?
Yeah, I mean, I think it's just too early for that. I mean, I think everybody sort of theoretically wants to know their status. I know I certainly want to, but the issue is like once you get that answer from one of these tests, is really what are you going to do with that data? And I think the point is number one, you sort of can't trust the results. I mean, they're useful to some degree, but you're going to misdiagnose a lot of people. But also, especially with these qualitative tests, we have no idea what level of antibody you have in your blood, and also more importantly, we have no idea if that actually protects from reinfection. And multiple agencies have come out sort of supporting this. This isn't just sort of my opinion. I mean, the WHO even made a big point, this was a couple of days ago, saying there's absolutely no evidence that you can just get infected once with this virus. I mean, none of this has been figured out to date. And these qualitative tests, really, the way that we're going to use these in society is mainly just to track spread sort of on an epidemiologic viewpoint, sort of broad brushstrokes. These tests should really not be used at all to guide any policy decisions. So that can be sort of on a governmental level as far as policies, but even locally or on a sort of personal level, even if you test positive with one of these tests, you still are required to wear your PPE in the office. And the antibodies that you have by no means will prevent future infection.
So that led right into my next question. Even the quantitative test, you don't feel it acts as like an immunity passport or a risk-free certificate that allows you to travel or return to work or not wear a mask or change your behaviors?
I think at some point it will, but right now, none of that's been truly worked out. I mean, I think in the future, and I think this is going to be 8 to 12, even more weeks probably before we figure this out. I think there will be a specific protein that will be sort of the sentinel protein that we look at, and we are going to build our immunity off of sort of quantitative levels against that protein. But that just has not been figured out. Even the ELISAs on the market are targeting different proteins of the virus, and we don't know which one's truly important for immunity. But, I mean, just for example, so I was talking to a colleague at Lilly about this issue, and they have a proprietary ELISA test, and they also have a group of employees that have been tested positive, and those are sort of their gold standard patients who've had PCR positivity and symptoms, and they're using their blood to sort of quantitate this. And on their ELISA, this guy was actually concerned because one of the things he's seeing is that at certain time points after infection, patients are not mounting really the antibody levels that you see with other diseases or other vaccines. So he was telling me that some of the early stuff that they have are showing most people are only mounting maybe 65% of the antibodies that they should have at a certain time point. And that sort of worries people because I think there's a concern that not only will we have low levels of antibodies, but they may actually fade over the next 6 months to a year and actually not provide long-term benefit. Now, we also may find as we track these patients that antibody levels really rise up and they provide protection, but all that has to be figured out, and it's just not there yet.
Well, that's even scarier when you look at the antibody response and the amount of antibody generated and our big hopes on a vaccine, because I think all of America is just banking on the fact that when a vaccine is developed, we're free and clear. But according to, what you just said, that might not be the case.
Yeah, I mean, I think that what the issue with the vaccine is they've got to find the right protein and target first. Now, antibody titers after a vaccine may be much higher, and we may be able to, design a vaccine in which we can get high titers that can provide protection. But at least there's some early evidence that if you get COVID or SARS-CoV-2, you personally may not mount, the right protective antibodies, and that's all preliminary data. But
interesting. Well, I've heard something about a saliva test from Rutgers. Tell me about that.
Yeah, I'm glad you brought that up. I think that was one of the interesting things that I saw about 7 or 10 days ago. So Rutgers in New Jersey was able to develop a test. Basically, the patient just kind of spits into a little test tube 2 or 3 times, and they were able to show that this test tested perfectly 100% compared to the nasopharyngeal swab. So it had the same specificity and sensitivity as that PCR. And I think people are excited about it because that could really help the people who are obtaining the test. They won't have to use as much PPE, and it probably will put them at much less risk if a patient just has to spit versus actually, sticking a swab down somebody's nose.
That
so
sounds
exciting.
Promising. Yeah, totally. How about the, we can at-home test just about everything now. And in fact, fun fact, I just had my 40th birthday this weekend, and one of my presents from one of my dear friends was a COVID test. So stay tuned for that. But, there's all these, testing opportunities and in-home tests. Tell me about those and how the FDA looks at-home tests.
Yeah, for a while there, they were not wanting to approve any at-home testing, but they did finally, this was, I think, about 10 days ago, they did approve, again, with the emergency approval process, an in-home test. LabCorp is the one that's providing this, and they're kind of selling it direct. It sounds like it's $119 per kit, and they say they have about 60,000 at-home kits available. And then basically all you have to have though is access to one of the LabCorp boxes. I mean, somehow it's got to get back to LabCorp. So a lot of times that may go through the doctor's office, but and then they run it on the same equipment that they normally would run. So it's
Are those
interesting.
swabs, Gus? Are those
those are
swabs?
Yep, those are nasopharyngeal swabs. Yep. So
I would think
haven't
that
seen
would
that.
be dangerous. Yeah.
Yeah, I haven't seen the actual package or how it works, but I assume there's a lot of instructions on there to sort of try to teach someone at home how to do it.
I know there have been some reports with just the healthcare workers swabbing patients and some reports of CSF leaks. So I would think that would be a tricky deal at home.
Yeah, that sounds concerning.
Let's talk about the LA. I know they were kind of cutting edge on getting antibody testing started. Tell me what they've found.
Yeah, so I mean that's really where when people talk about these antibody tests, I mean they're mainly going to be used for sort of patient tracking, and some of the preliminary data out of LA and its main county there is showing a high sort of positivity rate. So they're seeing about 40 times the number of patients have probably been exposed to this virus as they originally thought. So I think that's, interesting. I mean, everybody talks about what kind of asymptomatic cases are out there, and there's various estimates. The CDC has sort of come to the idea that maybe 25% of patients are asymptomatic, but we don't really know how many patients are sort of mildly symptomatic either. But it looks like a lot based on some of this preliminary data from LA, and I bet you in other big cities we'll also see that. But as the city size and density probably decreases, we'll probably see less of that total denominator.
Well, that's great. I feel much more educated on the antibody test. Any summaries for Forefront Derms, PAs, and NPs on this overall testing that you want to leave everyone with?
Yeah, I guess I would just say, I mean, everybody's really excited about this testing. I know I personally want to know my test results, but right now I'm not sure we don't know which manufacturers are great and which are not, and they're expensive tests. And we don't know the sensitivity and specificity, so I think it's best if we all just hold on and wait for better testing to come out, and just wait. All these questions are going to be answered soon, and we're going to have hopefully good tests, and we're going to have good recommendations on when you can sort of get your immunity passport and when you can sort of potentially start living your lives again. So I know I'm committed to sort of getting that data to Forefront. I'm sure everybody is anxious to get the results, but I want to make sure we get good information out there before we start just testing willy-nilly.
Awesome. Well, this has been Gus Natale, my first guest on the Forefront, what ended up being 15 this time, but a really great topic. Gus is double boarded in internal medicine and dermatology, extremely smart, has been instrumental in our task force and implementation of our protocols. So Gus, thanks for all you've done from all of the Forefront docs, PAs, and NPs, and thanks for being my first guest.
Yeah, thanks, Betsy. It was fun. I appreciate it.